SDS-PAGE stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis. It is an analytical technique to separate protein samples based on their molecular weight. Principle of SDS-PAGE states that a charged molecule migrates to the electrode with the opposite sign when placed in an electric field. The separation of the charged molecules depends upon the relative mobility of charged species. Smaller molecules migrate faster due to less resistance during electrophoresis. The structure and the charge of the proteins also influence the rate of migration. Sodium dodecyl sulfate and polyacrylamide eliminate the influence of structure and the charge of the proteins, and the proteins are separate based on the length of the polypeptide chain. The materials needed are power supplies, gels, electrophoresis chambers, protein samples, running buffer, staining and destaining buffer, and protein ladder. Firstly, gather the combs, glass plates, spacer or silicone tubing, and binder clips. A comb is used to make wells to load samples, so we should be mindful to use an appropriate comb depending on the sample size. For example, use an 8-length comb for 7 samples and molecular weight markers. Next, thoroughly clean the glass plates with ethanol and assemble the gel casting mold. After that, pour acrylamide solution for a separating gel. Overlay with water to prevent contact with air or oxygen, which inhibits polymerization. Then, allow acrylamide to polymerize for 40 minutes to form a gel. Proteins migrate at different rates depending on the concentration of the separating gel. Therefore, it is advised to use an appropriate gel concentration for our target protein. Using a higher concentration of acrylamide, we produce a gel with a smaller mesh size. This is suitable for the separation of small proteins. Therefore, in general, an acrylamide concentration between 6 to 15% will be used. Pour acrylamide solution for a stacking gel. Gently insert a comb parallel to a separating gel. Then, allow the acrylamide to polymerize. Proteins are highly concentrated when they migrate through a stacking gel prior to entering a separating gel. The concentration occurs due to the difference in the rate of migration of glycine ion, chloride ion, and proteins. After the acrylamide has polymerized and formed a separating gel and stacking gel, remove all the apparatus apart. Then, separate the separating and stacking gel plates by using a knife. Rinse each gel plate with water. Gently remove the comb with both hands slowly. Rinse again and our SDS-PAGE is prepared. Put the protein samples into a heat block. Heat the protein samples at 95 degrees Celsius for 5 minutes. Firstly, the comb is removed from the gel assembly. Then, the gel is mounted on the electrophoresis apparatus using binder clips. The running buffer is poured into the upper and lower chambers of the electrophoresis apparatus. The air bubbles and small pieces of gel should be removed from the wells and under the gel by using a shrink. After that, carefully take a sample and molecular weight marker and then load them into a well one by one. Close the lid of electrophoresis apparatus. Turn on the power supply and run the gel until the dye BPB in the sample buffer reach the bottom of the gel. When the sample buffer has reached the bottom of the gel, remove the gel assembly from the electrophoresis apparatus. Then, remove the gel from the glass plates by using a spatula. Now, the gel is ready for the further preparation of the subsequent analysis.